Good evening. I thank the scientific and organizational committee for this conference. My name is Souddi Mohammed, Associate Professor at Adrar University, Algeria. My presentation is titled Diversity of Weeds in Food Legume Cultivation in Hyper-Arid Area of Algeria. The legumes occupies an important place at the level of agrosystems, given their agronomic, economic, and nutritional interests. In Algeria, food legumes occupy third place in terms of area after cereals and fodder. In Oasis agrosystems, Vicia faba occupies an important place in the life of Wasian populations. Medicago sativa is a forage resource for livestock and also plays a significant role particularly in oasis. Agriculture production is severely limited by several factors including disease, insects, and weeds. Weed pressure is one of the factors that significantly reduce cultivation production. Weeds have a direct negative effect through competition with cultivation for the elements necessary for growth: water, nutrients, light, and development space. The work carried out on agrosystems weeds in Sahara zone located in northern Sahara, Syed 2009, Syed et al. 2014, Edud et al. 2018. In this context, the objective of this work is to study the diversity of weeds in food legume cultivation in Saharan agrosystems. The present study was carried out in the oasis of Tamantit, which is located in southwest of Algeria. The municipality of Tamantit is located 12 kilometers south of the town of Adrar. The climate of the study area is hyper-arid, characterized by very high temperatures in summer and cold winter. Rainfall is low and irregular. Methodology. The cultivation system practiced has two to three strata: palm trees, fruit trees, and low cultivation, which are generally cereals, legumes, fodder crops, condiments, and vegetable crops. The inventories were carried out in spring of 2022. The study of weeds in field legumes focused on 10 phytoecological releases carried out in the two cultivation: the cultivation of Medicago sativa 5 releases and Vicia faba 5 releases. The floristic releases are carried out on homogeneous and representative surface of 1 square meter. In each release, species name and number of individuals of each species were determined. Species identification was carried out using different flora: Kizel and Santa 1962-1963, Ozenda 2004. The determination of the biological types of the listed species is based on the work of Kizel and Santa 1962-1963 and Kizel 1965 to establish the raw and real biological spectrum of each type of culture and the study area. The morphological types annual and periannual were determined from the work of Kizel and Santa 1962-1963 and Ozenda 2004. Data analysis. The quantitative study of width encountered is based on the analysis of density and frequency. Result and discussion. The weed flora of wood legumes includes 23 species divided into 21 genera belonging to 5 botanical families. The number of weeds recorded in legume cultivation can be related to climatic conditions favorable to the development of these weeds. According to the type of cultivation, Vicia faba is the richest with 17 species, 16 genera, and 12 families. Medicago sativa is the least rich cultivation with 15 species, 14 genera, and 10 families. The variation in floristic richness between the two cultivations depends on the use or absence of herbicides. Biological spectrum. In two cultivation and at the study area level, pterophytes are the most represented biological type regardless of the raw or real spectra with percentage exceeding 17%. The dominance of pterophytes results from their adaptations to the condition of the agrosystem and water availability. Morphological spectrum. The raw and real morphological spectrum of two cultivation and at the level of the study area indicate a good representation of annual species compared to perennial species with value exceeding 18%. The proliferation of annual species in the environment could be explained by climatic factors such as wind, water, or animals. The presence of perennial species is due to the absence of herbicide treatments. Phytogeographic spectrum. The raw and real phytogeographical spectrum of two cultivations and at the level of the study area indicate a good representation of the cosmopolitan element. The dominance of cosmopolitan species is explained by the fact that the cultural biotope is a very open environment. The Mediterranean element comes in second position at the level of the raw and real spectrum of the study area. This element occupies the same place in the cultivation of Medicago sativa and Vicia faba. This reflects the global affinity of our flora with the Mediterranean region and its climatic conditions. Importance of weeds. To understand the importance of weeds and develop appropriate program to reduce their damage in food legume cultivations, the density and frequency of each species were calculated. Anagalis arvensis, Chonsus olivaceus, and Imex spinosa species represent the highest densities. Anagalis arvensis recorded the highest density with 4.3 plant per square meter, while the density of Chonsus olivaceus is 3.1 plant per square meter, followed by Imex spinosa with a density of 2.6 plant per square meter. The analysis of the frequency of species revealed the existence of 6 classes. Plants with a frequency between 75 and 100% are the most infesting species, with frequencies exceeding 75%. Anagalis arvensis and Sonchus oligaceus are the two most infesting species. Conclusion. This work aims to study the floristic diversity of weeds that cause the domination of the production of food legumes in waste agrosystems. Through 10 phytoecological releases, we were able to identify 23 weed species represented mainly by annual species. Anagalis arvensis, Sonchus oligaceus, and Imex spinosa are the most frequent and abundant species. The dominant biological type is pterophytes, which complete their development cycle during the good season, which coincides with the cultivation irrigation period. Phytogeographically, the most abundant plants are cosmopolitan species that bear witness to human activity. The results obtained constitute an important source of information for the management of agrosystems in hyper-arid zones. Thank you.